Go with the mate.
Okay, let me share. So it's 9 a.m. Pacific time, and I'm delighted to be opening another Global IMMUNOTALK. I'm here with one of the co-organizers, Dr. Carla Rodley, and our fantastic speaker today, Dr. Ana Domingos. So Carla will introduce Ana now. Thank you.
Yes, thank you, Elena, and welcome everybody to a new Global IMMUNOTALK. It is a real pleasure for me to introduce our speaker today, Dr. Ana Domingos. Ana is an associate professor of neuroscience at Oxford University. Now, Ana, as many of our speakers, is a true international scientist. She was born in Portugal. And after her undergrad, she came to the United States, where she did her PhD studies at Rockefeller University with Dr. Leslie Boshall. And it was during this time that she learned about the power of employing transgenic lines in combination with optogenetics. Now, I think this is something that Ana has nicely applied, particularly during her postdoctoral studies, which were also at Rockefeller, but with Jeff Friedman. And during her postdoc, she discovered that she could trick mice to drink water flavored with artificial sweetener by stimulating the release of dopamine from neurons in the reward system with light. So using these transgenic approaches and these optogenetic approaches. After her postdoc, she moved in 2013 to Portugal, to the Gulbenkian Institute to start her own independent laboratory. And more recently, as I mentioned, she moved to Oxford, where she's an associate professor. Since then, she has focused on defining the neuroimmune mechanisms that underlie obesity. And her laboratory discovered interconnections between neurons, adipocytes, and macrophages that regulate fat mass. Specifically, her studies have shown the central role of macrophages that are associated with sympathetic neurons in the metabolism of noradrenaline. And this has led her to pursue the development of drugs that can target the sympathetic axis in the periphery and allowing, to mitigate in this way obesity while sparing adverse effects of sympathomimetics that are more at the central level. Now, Ana has received support from prestigious organizations and is currently an HHMI Wellcome International Research Scholar. I would also like to mention that in a relatively short time, she has become a thought leader in neuroimmunometabolism. Ana, again, it's a real pleasure to host you today. Thank you so much for accepting our invitation.
Thank you so much, Carla. Can you hear me well?
Thank you. We can hear you very well. So we should mention, we do this live, and if you probably noticed that we were a little bit nervous, it's because we've been having one problem, which is that we cannot get to see Ana. We don't have her camera working, but we can get to listen to her, and we'll get to know about her wonderful research, and we'll get to see her slides. So everything should still be okay, and we're absolutely delighted to have you here, Ana. Even if we cannot see you, we know you're there.
Yeah, this is going to be like the good old radio days.
That sounds great. We can all adapt and still, communicate.
Yeah, I don't know why the camera is not working. I mean, it was working yesterday. So thank you so much for the very comprehensive introduction.
Yes,
this is really, I'm touched by your very comprehensive introduction that spans a big portion of my life, starting with the postdoc and my PhD. So I'm going to talk about...
sorry, go ahead, Ana, go ahead.
No, I was going to start my talk by, is the slide sharing okay? You see the slides?
So not yet, but we have time to do that. So let's see, let's start to see if you can share your slides, but...
I thought I was already sharing them. Okay, so apparently I'm not.
They're not. But
Okay.
don't worry about it, because even before we start learning about your beautiful discoveries, once you are able, let's see, I don't want to interrupt you if you are trying to share the slides. Let's see. Let's share your slide first.
Is it okay? Let me see.
That is fantastic, and you can put the presentation. Let's see.
Okay. So the presentation.
The presentation mode.
Where's my cursor?
Take your time.
My cursor.
There you are. Yeah.
There you are. Is my cursor?
Yes, we can see your cursor. You see it there? Perfect.
Okay. Here we go. Can you see my slide?
We see your first slide very, very well. But as you know, there's something very special about this talk, which is that we love to learn more about our speakers. And, you know, you have been really an emerging leader in neuroimmune metabolism. And so before we start with your talk, and now we're all under control, your slide is showing, we would really love to know more from you. We would love to know whether you can share with us some of the highlights and also the challenges that you have encountered in helping pave the way of this new field, of this neuroimmunometabolism field. So before we start, can we learn from you? What are the highlights and challenges of this path that you have taken?
There's also, beyond challenges, there's also positive aspects and pleasure that derives from doing discovery. And what happens many times throughout scientific discovery is that when you have an intersection of fields and you have a person from one field stepping into another field, that person steps into that other field with new eyes, with a new perspective that those who are already living in that field would not have, or would have a lower chance of having. So I think it's a sword with two edges, because you may have original ideas that are important from different fields, but you're also a rare bird. Let's put it this way. Whenever there's a person that it's from a different field that tries to step into a field. So I think maybe that's the biggest challenge of doing cross-disciplinary research, is that everybody speaks their own language and somebody has to speak both languages. Mm-hmm. And in order to speak two languages, it's very, very difficult to be perfectly bilingual in two languages, let alone three languages, right? So I think that's maybe the challenge, that there's this human behavior of owning a language and being very proficient in that language. And then if you try to learn a new language, then there's going to be barriers and there's going to be challenges. But so far I've been very lucky, because I was trained as a neuroscientist and the immunology community has been very, very warm to me. So I have nothing but to thank the immunology community. They invite me for lots of meetings. So I think it's a movie with a happy ending, or a happy beginning, hopefully. That the challenges that a lot of people face, and I face myself, because I started off as a mathematician before I did it. I did molecular biology in Drosophila with Leslie, right? So there was throughout my life, I always had this constant need to learn new languages, need to learn new things, and try to come up with something unexpected in a given domain, because I get really, really bored of going down the beaten path. Right. But that's just a personal aspect. Of course, funding agencies, they like the beaten path. So that's another challenge. That's one of the challenges. Right? Because it's one person alone against a group, and most of the times the majorities win, right?
Yeah.
So sometimes I hope I said enough.
Yeah, no. Thank you so much for sharing that. And we really appreciate the fresh perspective that you bring to this field. And we are all ears. And yeah, we look forward to your talk. Thank you.
I am really touched by your initiative and also your introduction. I think it was one of the best introductions I've ever heard. So I'm really touched by your attention to detail and thorough work and effort that you put into your own initiatives. So your congratulations.
Thank you. No, thank you, Anna. Thank you. So it's lovely to have you. Okay, we are ready for your talk. Thank you so much.
Okay, I already gave all the thanks I wanted to give, and let's talk about neuroimmunometabolism. So neuroimmunometabolism here, it's tricolor because it represents each one of the colored cell types that you see in this image. We have macrophages, we have neurons, which are here the red spaghetti, so to speak, and we have the adipocytes, which are the cells that constitute what we call our fats, our adipose tissue. And the way I use the word metabolism here is really to represent the homeostasis, the body weight of homeostasis. So whether you get obese or not, right? So this is the way I use the word metabolism, because the word metabolism can mean something else inside a cell. And here we're talking about a different zoom level, a different level of organization, which is the level of organization of the whole individual, right? So this is what I talk about metabolism. And one key piece in this puzzle in the control of the body weight homeostasis is a hormone named leptin that it's produced by the adipose tissue. And it's produced in amounts that are proportional to the amount of fat. And these are the landmark papers from Jeffrey Friedman's lab and also Steve O'Reilly lab, demonstrating that the adipose tissue produces this hormone, and that the hormone signals in the brain to inform the brain of how much fat exists in the body. And a very acute effect that one sees upon leptin treatment of individuals and animals that do not have the gene that encodes for leptin, so who are leptin deficient. The very pronounced effect one sees from leptin rescue in leptin deficient models is the suppression of food intake. So that's really, really evident. It's measurable. It's easy to measure. And for a long time, people think that the neuroendocrine loop of leptin action, and here there's also another element, is that the adipose tissue, it's not just a tissue that you have in a body to protect you against shock. It's a tissue that it's in fact an endocrine gland because it produces hormones, and hormones, they usually have neuroendocrine cycles. So here we're looking at the neuroendocrine cycle of leptin action in the brain. And for a long time, it was thought that the sole efferent axis, so the axis that comes down from the brain, as opposed from the afferent axis, which is leptin itself, for a long time, it was thought that suppression of food intake was sufficient as an efferent axis. Of course, then the clock ticks the other way around. When the fat tissue is small, there's less leptin. That leads to an increase of food intake, if that alone is sufficient to reduce fat mass, and so on, and to increase fat mass. So this idea that the efferent, so the motor element of this neuroendocrine loop that ticks like the clock, was food intake, has been denied by studies in which we pair feeds leptin deficient animals to those who are receiving the treatment. So I get treated for leptin, being leptin deficient, I lose weight, and then you are going to have the same amount of food that I eat. And they saw that animals who receive the same amount of food as leptin treated animals, they don't lose the same amount of weight. So there's a delta here, as you can see here. They just don't catch up to the weight loss that one sees in animals that have been rescued of leptin. And it's not just one group that saw that. There's another study who saw the same, and we saw the same in our own lab. So what could be the mediator of this delta here? What could be driving the weight loss that goes beyond suppression of food intake? So that's the question that has teased me ever since I started my own lab. And one observation that drew my attention was that leptin deficient patients, they have this clinical state in which they have orthostatic hypotension. What does that mean? It means that when you lift up the couch or lift up from a chair, your blood pressure is going to go low suddenly. And normal people don't have that. Normal people, you shouldn't see significant changes in blood pressure if you go from sitting to standing. And that is thought to be mediated by nerves that innervate the vasculature, the sympathetic nerves that innervate the vasculature. Also, there's another test, which is the cold pressure response test, which is again a readout in terms of blood pressure, in which you just stick your hand into a bucket full of ice, and then on the other arm, you're going to measure blood pressure using a cuff, an arm cuff. And there's deficient responses in this test as well. Although patients, they have normal blood pressure, so they're not hypertensive like the majority of obese patients. They have the tendency to be hypotensive. Now, this led me to think that maybe there's a deficient activity in sympathetic neurons, but not the ones that go to the vasculature that would then lead into a readout in terms of blood pressure, but those that would go to the adipose tissue. Now, the question is, are there any sympathetic neurons in the adipose tissue? And at the time when I was asking these questions to myself, I read all the books that you can imagine in terms of neuroanatomy, and this is a German book. Of course, I don't read German, but these books, the cartoon is the same in every single diagram. And the adipose tissue was simply not represented. The brown adipose tissue, which is a tissue that drives thermogenesis, that makes you warm, essential, and it burns fat by keeping you warm, or it keeps you warm by keep burning fat, was known to be innervated by the sympathetic nerve. But the question I was asking is it the case that the white adipose tissue, which produces leptin, was in fact innervated? And there was just no answer for that, because some people would say yes, some people would say no, but the primary data was simply not convincing. There were no convincing pictures of adipose tissue that was sliced, and you could see neurons there. So it took a change of techniques in order to see something. And the technique that we took advantage was a technique that was being developed by several groups, Halli Herturk in Munich, also Karl Roth, Clarity, mostly being applied to the brain. But we apply these clearing techniques in which you have a piece of tissue. This is a piece of inguinal adipose tissue. You run it through a series of chemical steps, organic solvents, so that you dilute out the fat, and then you have a piece of transparent adipose tissue. And with the transparent adipose tissue, we could finally see what we couldn't see before. Let me see if the movie rotates. Okay, there we are. So as you can see here, this was a piece of transparent adipose tissue that was stained against TH, which is tyrosine hydroxylase, which is an enzyme in the biosynthetic pathway for catecholamines. And as you can see here, there's a generous tree of neurons that are innervating the white adipose tissue. Now, this is convincing. These are neurons, and I'm a neuroscientist. No neuroscientists would not say that these are not neurons. You can imagine that if you slice up this image, you'll get dots. And then, of course, the dots are not so convincing. But with this type of technique, we can convincingly we can be convinced that sympathetic neurons are present in the adipose tissue. Now, the question is, are these neurons just subserving the biology of the blood vessels that also exist in this tissue, or they're also actually making contacts with the adipose tissue? And for that, we had to use another technique, which is to photon microscopy, in which we had a living animal under the microscope, and using transgenic mice, which render those neurons fluorescent by expression of a fluorescent reporter named tomato, which is a GFP variant. And it expresses exclusively in neurons that express the marker that I just stained before, TH. So then we can see that, in fact, there's close contacts between adipocytes and these neurons. Now, we knew from textbooks that these neurons, they produce norepinephrine. And we also know from textbooks that norepinephrine activates the adrenergic receptor, and that downstream of the adrenergic receptor in adipocytes, there's lipolysis. And textbooks also tell us that these green cells, the adipocytes, they express adrenergic receptors. Mice express beta-3, and humans, they may express more than beta-3 other receptors. So downstream of this activation, there's lipolysis. Now, we knew all of these pieces of the puzzle. What we didn't know is whether the source of norepinephrine that underlies homeostatic changes in body weight and adiposity are these neurons, or something else, for instance, the adrenal gland. The adrenal gland also produces norepinephrine, and that usually happens if you have to run away from the lion or a snake or a spider, a big furry spider. So that's called the fight or flight response. Adrenal glands, they're not necessary for body weight homeostasis. When patients or animals get their adrenal glands removed, they still can respond to homeostatic changes in body weight. So for instance, they will lose weight upon leptin injection. So if it's not the adrenal glands, what is it? Are these the neurons that are causal to body weight homeostasis? So this is a question that bodes for a gain-of-function experiment in which we utilize the modern tools of neuroscience, which is optogenetics. Optogenetics, they've been popularized mostly by Carl Neisser-Roth at Stanford. And we expressed channel rhodopsin in sympathetic neurons. And after that, we validated that it's expressed in sympathetic neurons and not in other neurons. And after that, we implanted an optical fiber deep down into the inguinal fat pad, only on one side, and left the other side as a contralateral control for controlling for systemic phenomena, for instance. We saw and we measured that the content of norepinephrine was higher if mice would be stimulated with light therapy. So channel rhodopsin is sensitive to light, and light drives activation of these neurons. And when norepinephrine is in excess in the adipose tissue, it drives what it's known to drive for, which is lipolysis, which is marked here with phospho-HSL, which is a biochemical marker. And these experiments were done by collaborator Wenwen Zhang. So these effects we see only if animals have channel rhodopsin, but not in animals that do not have channel rhodopsin, but were subject to the same experimental procedure. In the long term, mice will lose fat locally in the site that was stimulated, but not in the site that was stimulated. And again, this is only seen in animals that have channel rhodopsin, but not in animals that do not have channel rhodopsin, but were subject to the same experimental procedure. This was a gain-of-function experiment that demonstrated that local activation of these neurons drives lipolysis in vivo locally. And the loss-of-function experiment is the reciprocal. So if we cut off the axons, either mechanically or using chemical tools or chemical stereotoxin, so the conditional tissue-specific ablation of neurons. So there's a lot more neurons in these nerve bundles that we're able to dissect that are not just sympathetic, and we're not touching those, we're touching the sympathetic neurons. And if we obliterate the activity of these neurons, then we suppress the effect on lipolysis, which is driven by leptin. More systemically, if we generically sympathectomize mice, we see that there's a very pronounced effect on obesity. So if we push these animals to become obese, they get obese really, really fast. Usually it takes 12 weeks to get an animal fat, but in this case, it took a few days to get them fat. And what's even more interesting is that in the absence of these neurons, if we put them on a diet and we stop the fattening stimulus, which is a high-fat diet, and we move them onto a normal, healthy diet, they just don't lose the weight. They will not lose it. So they will stay chubby for the rest of their lives. And this is all independent of food intake. So what this tells us is that the efferent arm of the neuroendocrine loop of leptin action are the sympathetic neurons that extend the action of leptin beyond food intake. And therefore, they close the neuroendocrine loop of leptin action in the brain. And I think now we have a complete picture of the neuroendocrine loop in which we have an afferent axis, which is leptin itself, and the efferent axis, which are the sympathetic neurons. And this is how the clock ticks in homeostasis. So like many neuroendocrine loops, you have an efferent signal that signals onto an organ, and then we have a descending efferent signal that signals back onto the gland where it was produced in the negative feedback fashion. Now, what happens during obesity in humans and in animals that are not leptin deficient is that there's an excess of this hormone being produced. So that's called hyperleptinemia. And hyperleptinemia leads to leptin resistance. And upon leptin resistance, for some reason, the brain is not sensitive to the action of leptin anymore. That downregulates the efferent signal. Now, the question is, would there be a way of overcoming or shortcutting central leptin resistance and restoring the feedback loop to its normal balance, in which the efferent arm would be just of the same thickness as or the same strength or amplitude as the afferent arm? So that's the question that my lab was interested ever since I started my lab, essentially, or ever since we discovered these neurons. Now, one way to shortcut leptin resistance would be exactly to thicken up or to amplify. This is a megaphone, so this is to amplify the efferent signal by facilitating the activity that has been suppressed by lack of leptin action. Now, of course, these neurons, if we would able to facilitate them, then they would zap the fat in the white adipose tissue and perhaps restore the neuroendocrine loop and make it tick in the other direction. Now, either an idea would be to do something to these neurons themselves, but maybe these neurons are also talking to other cells. And instead of targeting the neurons directly, I asked whether or not it would be possible to do this via another cell. And to do it via another cell would just have another element of control that in terms of translation to pharmacotherapies would be desirable. Now, the cells that I'm talking about were cells which are macrophages, that now we know are macrophages, that were sitting on top of these nerves. And the temptation to study was very high because these cells, they really look beautiful. And to decide to study something because of its beauty, I think it's a good hunch to have. And I recommend my students to follow their hearts and to follow what they think is pretty. And definitely these cells were very pretty to look at, and they merit study. So what we did was to isolate the cells by flow cytometry and compare them to all the neighboring cells and also cells that are very studied, such as the splenocytes and the microglia. And we saw indeed that the sympathetic associated macrophages that were dissected, that were isolated from dissected nerve fibers in the inguinal fat, in terms of gene expression, in looking at the principal component analysis, which was done by Jason Seidman in the Chris Glass lab at UCSD, we clearly see that they stand alone. So they have their own cluster. They're very different from microglia, even though they are associated to nerves. And this is a question that I got a lot. How do I know that these cells are not microglia? Well, they're outside of the brain. And moreover, their gene expression profiles are very, very different from microglia. They're very different from splenocytes, and they're sufficiently different from neighboring cells. It's just adjacent to them, which are the adipose tissue macrophages. Zooming in into the gene expression profiles, we realize that there are genes that seem to be pan-myeloid, like the beta-2 adrenergic receptor that we see here in this blue bar. It seems to be expressed quite heavily in all macrophages that we came across. Now, there were genes that were statistically enriched in SAMs, and here we highlight one, that's the norepinephrine transporter, solid carrier 682, which is a name that is not as palatable as norepinephrine transporter. So we decided to study whether or not this gene had any function in macrophages. And what we're looking at here is basically a superior cervical ganglia that contains neurons in red and the macrophages in green dancing on top of these neurons. And we made this preparation where we could study ex vivo the function of these macrophages. This is how a superior cervical ganglia looks like. It's a collection of neurons and also macrophages. And we thought that because these macrophages were dancing on top of the neurons, chances are that the macrophages are going to be sensitive to the activity of neurons. So neurons are electroexcitable cells, and we decided to excite these cells again with optogenetics to see if there was any reaction coming from the macrophages. And the reaction we saw was that there was an increased content of norepinephrine, which is the neurotransmitter of the neurons, inside of the cells. And this happened mostly if there was a sensitivity to the light stimulation given by channel rhodopsin relative to controls that did not have channel rhodopsin. Now, if the cells are accumulating norepinephrine inside of them, there's only two ways this can happen. Either the cells are making the norepinephrine themselves, or they're taking up norepinephrine from the outside world. And there were several groups at the time, or even 10 years before, suggesting that macrophages can produce norepinephrine, but we didn't see that. So these cells, they don't have the machinery for making norepinephrine, not SAMs, not any other adipose tissue macrophage that we came across. And other groups have published the same thing. So these cells, they do not have the biosynthetic capacity for making catecholamines, but they do have the machinery for taking up catecholamines. So this is, again, a different type, a revalidation of what we saw in RNA-seq data by qPCR. And they also have the machinery to degrade norepinephrine. This presentation will focus on the transport, will not focus so much on the monoamine oxidase, which has been studied by the group of Vishwadip Dixit, and I will refer to those studies in a bit. So if it is the case that the transporter is doing the job, then if we block the transporter, then we shouldn't see any norepinephrine inside of the cells, and that's exactly what happened. And if the norepinephrine is not inside of the cells, then it's outside of the cells in the extracellular space. So you can think about these cells as little vacuum cleaners that limit the extracellular availability of norepinephrine even during tissue homeostasis. So this is a good thing to have. The tissue during homeostasis protects itself and keeps it clean and functional by having these little vacuum cleaners that go around and clean up the excesses, let's put it this way. Now, all this is related to obesity. We realized that we ran the experiment with two different mouse models of obesity, one that is genetically obese, another one that is behaviorally induced. So this is a high-fat diet obesity model. We saw that there was an excessive amount of these vacuum cleaners, excessive amount of SAMs in the nerve fibers that we were dissecting from fat pads. And that in percent-wise, that excess was even higher than what the excess that is usually seen in adipose tissue. And the spleen doesn't really care about obesity. So we thought that perhaps if there's an increase of SAMs in sympathetic nerves, could we possibly revert obesity by performing a loss of function of SAMs? And we did that using the tools that immunologists use very often. So we had to learn from them. And what we did was to do a bone marrow transfer of a norepinephrine transporter, a whole body knockout, onto an obese model. Now, one may criticize that this approach is not tissue specific, but in my defense, in my lab defense, I would say that it's sufficiently tissue specific because no other cell in the hematopoietic lineage seems to express the norepinephrine transporter. That's what databases say. That's what we saw also by doing validations in the lab using the tissues of these animals. So these manipulations seem to be sufficiently tissue specific for the cells that we wanted to study. And doing this, so we have these obese mice that have a norepinephrine knockout from the bone marrow. We immediately saw that these animals, they had more brown adipose tissue. As I mentioned before, brown adipose tissue is a tissue that generates heat, that burns fuel by generating heat. OB mice, they're usually hypothermic. They don't like cold. They don't do well with cold. And they also have a whitened brown adipose tissue, meaning they don't really have a brown adipose tissue. If you try to look for it, you just see white fat. However, these chimeras, they seem to have that brown adipose tissue rescues. And the one thing we know is that norepinephrine promotes browning to the extent that not only the brown adipose tissue, or in the anatomical location where you usually found the brown adipose tissue, there was brown adipose tissue to be seen in these chimeras, but also the white adipose tissue now had been browned. So there was more multilocular brown adipose tissue cells, even in the white adipose tissue. And functionally, we also saw the impact of having a brown adipose tissue. So OB mice, they're not just obese, but they also are, they don't like cold, they succumb to cold. But these animals, these chimeras that had the different bone marrow, they could survive the cold a lot better. So they could do thermogenesis. So this is functional brown adipose tissue that was in fact burning calories under cold stimulation. Another challenge we did to these mice, so again, perturbing homeostasis, because this is all important to keep homeostasis, was to food restrict the mice for a period of time. And this is not food restriction that the animals don't eat. This is just normalizing their food intake to that of a normal mouse. Usually mice, they eat 3 grams a day. OB mice, they're hyperphagic and they eat about 6 grams a day. Now, between 5 and 6. What we did was just to normalize their food intake to that of a normal mouse. And the animals, the chimeras, they responded a lot better to that normalization, again, because they're not trying to fight off weight loss. And the rebound effect, or the yo-yo effect that most patients and animals for sure seen here experience after a period of food normalization that goes below their homeostatic set point, is that they shoot up. So there's a rebound effect on food intake and they gain all the weight that they have lost. This is called the yo-yo effect. The yo-yo effect is gone in the chimeras. So they have a long lasting, and this is 16 weeks. So this is very long lasting. This is not a short term period manipulation. These animals, they have lost their weight permanently. And again, this shakes up a bit the concepts because all of the homeostasis has been centered in the brain. And I think that if we have a neuroendocrine loop that has two components, and if these components are connected, we could just might as well change the functioning of the circuit by affecting one of the nodes of the circuit, not necessarily the brain. Also because it's very difficult to deliver drugs to the brain. So why not to try and challenge that notion? Again, so this confirmed that macrophages can dispose of catecholamines, and this is a very nice review by Michael Check. And three independent groups, they converged onto that same idea in 2017, acknowledging that macrophages can be mediators of norepinephrine catabolism. To make a long story short, we have sympathetic associated macrophages that attach to sympathetic nerves to keep the homeostasis of the sympathetic nervous system. In a situation of obesity, there's a lot more of these cells mopping up the signal that the adipocytes need to see in order to shrink. And if we do a genetic manipulation in which we abrogate the function of the norepinephrine transporter from the hematopoietic lineage, and this is, again, it's a manipulation that it's outside of the brain, completely outside of the brain, then we can restore homeostasis and we can force animals to lose weight. Now, of course, we would like to phenocopy this effect with a drug, basically a drug that would mimic the effect of the cork here, that it's clogging up the hose of the vacuum cleaner. And there's a family, big families of drugs that do this. This is the norepinephrine transporter, and drugs in the amphetamine class, they do that. So amphetamines, they enter the brain and they have a very potent anorexigenic effect. That is, they suppress appetite. And they also have a hyperkinetic effect, which means that they make people run around or dance all night long. Now, these drugs are also named as sympathomimetics, particularly indirect sympathomimetics, because they clog up the transporters and they create an abundance of the neurotransmitter that sympathetic neurons that sympathetic neurons produce, which is norepinephrine. That it's not to say that sympathomimetic drugs is an equivalent of drugs that activate sympathetic nervous system directly. There is no evidence that these drugs are able to activate sympathetic neurons directly. And because of that lack of evidence, we've run the experiment ourselves. We dissociated sympathetic neurons from superior cervical ganglia. They're happily living on a dish, and they look like this. They're actually very beautiful neurons. And by using electrophysiology, we could listen to the activity of the neurons. And these neurons, in the presence of amphetamine, they don't fire. They will not fire unless we provide, or they will not fire more unless we provide an incoming stimulus. In a PEGy configuration, we're holding the membrane potential of the cell, and the amphetamine does not overcome that handle. It's not until we start injecting pulses of current that then we see that there's more action potentials per step of injection of current. So we need to drive the cells in order to see the effect of amphetamine. That it's not to say that amphetamine activates sympathetic neurons. That is to say that amphetamine facilitates the activity of the sympathetic neurons. Those are completely two different, or not completely different, but maybe it's a subtle difference, but it does make a difference for the points that I'm trying to make here. So what we thought was that could we chemically modify amphetamine so that it doesn't enter the brain, and instead it has a peripheral action that could perhaps phenocopy the effect or the genetic effects that we saw. This was our initial hypothesis. This is what we had in mind. And we could not make big predictions about whether or not this molecule would bind the norepinephrine transporter, simply because the crystal structure of the norepinephrine transporter was not available at the time when we were making these studies. And I don't think it's still available. So in order to make good predictions about how drugs should bind a receptor, we need to have the crystal structure, preferentially the crystal structure co-crystallized with its natural ligand. But we didn't have that luxury. So it was cheap enough to just make the molecule and test it in rodents and see what happens. So sometimes that's also a good attitude, the let's do attitude, to take in research rather than going down the beaten path. And we were able to see that the chemical modification that we did indeed abided to our initial hypothesis that this doesn't enter the brain. And the rule of thumb is that if a molecule is big and is polar, it's not going to enter the brain. So brain, they usually like small molecules, like amphetamine itself. It very avidly goes into the brain. And we see here on a mass spec result of egg brain extracts of perfused animals, that the theoretical peak in red, it actually overlaps with the real chemical peak of the molecule. So amphetamine is present in the brain, but if we inject pegylated amphetamine systemically, we see the theoretical peak, but we don't see the real peak. So we don't see pegylated amphetamine in the brains of these animals. And what further substantiates this absence of pegylated amphetamine in the brain is that whereas animals that take amphetamine, they run around like crazy. Here we see the tracks of animals that were injected with amphetamine. The pegylated amphetamine mice, they don't really move as much as they move like controls essentially. And we see the same thing about in food intake. So animals that were injected with pegylated amphetamine, they don't really have an anorexigenic effect, which is typical of amphetamine prescription. The sympathofacilitator effect that we described early on for amphetamine seems to be preserved. So we have a biological effect in vitro that is preserved despite the chemical modification. And we see that with electrophysiology. And we also see that with calcium imaging. Calcium imaging enables us to drive the neurons with the endogenous ligand of the sympathetic neurons, or the endogenous driver, which is acetylcholine, which is produced by the spinal cord that plugs onto sympathetic ganglia. So the pre-sympathetic input. And here we see that when we drive these neurons with a puff of acetylcholine, that happens here in the second at 35 seconds, there's a facilitation of this response in the presence of either compound, and that's summarized here and quantified. Now, if we prescribe this either drug to animals that are either getting fat or that are trying to lose weight once they're already obese, we see that there's a weight loss with a size effect that is comparable. But what's going on here? Because the amphetamine animals, they're amphetamine-treated animals, they're running more, eating less, but the energetics tells us that the math doesn't add up, right? Because pegylated amphetamine animals, they're losing weight, but they're not eating less or running more. So what's going on here? Where are they burning their fuel? They're burning the fuel because they're engaging thermogenesis, but the amphetamine animals are also engaged in thermogenesis. So what's going on here? What's going on here is that the animals that are taking amphetamine, they're actually very sick because they are hyperthermic, whereas those who do not have, were injected with pegylated amphetamine, they have a normal core temperature. So if we probe the temperature, the core temperature of these animals, we see that pegylated amphetamine treated animals, they're normothermic, whereas those that are injected with amphetamine, they're hyperthermic to the point that they can get sick and die. And that sometimes happens to patients as well. And so how come they're producing more energy or producing more heat, and they're not getting, where are they getting rid of their heat? Pegylated treated animals are getting rid of the heat because they have vasodilation in the periphery, whereas amphetamine is a well-known vasoconstrictor. So essentially amphetamine animals, they are producing a lot of heat, but they're retaining the heat and they're getting sick and getting malaise with that. Whereas those, the pegylated amphetamine treated animals, they are producing heat, but they're getting rid of the heat. So the analogy here is that vasodilation works like a window where you let the heat dissipate. Now, a big concern with sympathomimetics is the cardiovascular effects. Most sympathomimetics, the great majority of them, they have notorious cardiovascular effects. They increase the blood pressure. They also drive tachycardia, and tachycardia in turn drives more higher blood pressure and also creates changes in the heart, in the structure of the heart, such as cardiac hyperplasia. and that's all very bad. But the pegylated amphetamine treated animals, they do not have that. And there was some indication from the literature that, in fact, again, this is one of the examples of cross-disciplinary research, that cardiologists, they would think that the effects of amphetamine, they happen directly in the heart, and therefore, if an amphetamine would facilitate the activity of sympathetic neurons, then the pegylated amphetamine should do the same. And it's known that sympathetic neurons, they drive the heart, so they have a chronotropic effect. But this is not happening here. And it's not happening here. And the only difference is that the pegylated amphetamine is not entering the brain. So if it's not entering the brain, is what the cardiologists think actually that it's a direct effect in the heart. It's not a direct effect in the heart, but instead an effect in the brain that in turn is driving the heart. And that's the case. So there was some evidence from the literature that amphetamines, and including methamphetamines also, they can drive the heart via central effects. And we just repeated the same experiment with our drugs, and we saw exactly the same. So amphetamines, when injected in the heart, they are sufficient to drive the heart, not the heart, when injected directly into the brain, they are sufficient to drive the heart. And same thing for the pegylated amphetamine. So by excluding the pegylated amphetamine from the brain, we are actually creating a cardioprotective effect, even though the drug is running around systemically. Now, lastly, because we didn't have the crystal structure of the norepinephrine transporter, and our assumption was that the mechanism or the molecular target of this drug was a norepinephrine transporter, because that was our departing point in terms of vision, in terms of hypothesis making, of building our hypothesis. And to finally know, or having a proof or disprove whether or not this drug was able to bind the norepinephrine transporter, we teamed up with a CRO in France who was able to run these radioligand competition assays, in which we have a high affinity ligand being bound to the norepinephrine transporter, and then we throw in our drug to see if it can outcompete with the radio labeled high affinity ligand. And whereas amphetamine is perfectly able to do that, then the pegylated amphetamine was not, which meant then that our initial hypothesis didn't have legs to walk. And that was kind of a disappointment, but that's the nature of research. Most times, the way you think, you have to go 180 degrees and go in the opposite direction. So we started looking for data that we already actually had in the drawer, that we didn't pay sufficient attention early on, which was that the sympathofacilitator effect disappears in the presence of a beta-2 adrenergic agonist blocker. So what does this tell us? It tells us that the sympathofacilitator effect is mediated by the beta-2 adrenergic receptor. Now, is it true that the pegylated amphetamine can bind to the receptor? Fortunately, we had available crystal structures of the beta-2 adrenergic receptor, co-crystallized with a natural ligand. And here we see perfectly well that the pegylated amphetamine, this is a simulation where we see the pegylated amphetamine bouncing around in the hydrophobic pocket of the beta-2 adrenergic receptor, even though it has lost its amine group. Even though it has lost its amine group and it has instead an amide group, there are interactions that are preserved, interactions that take place with the endogenous ligand, that this drug is still able to recapitulate. So the replacement of the amine by the imide group preserves the interactions within the hydrophobic pocket of the beta-2 adrenergic receptor. So the amine group, it seems that it's, after all, not necessary for every single interaction with every single receptor, and that in order to make any conclusions about the interactions between a drug and a receptor, it's better to have the crystal structure in hand. Unfortunately, for the beta-2, for the norepinephrine transporter, that was not available. Now, could we then use commonly prescribed drugs that are beta-2 adrenergic antagonists? Even propanol is one that it's used in the clinic. Could we use it as an antidote for the weight loss effect? And the answer is yes. So by blocking the beta-2 adrenergic receptor, we can mediate the weight loss that it's seen by prescription of the pegylated amphetamine, and everything happens in the absence of food intake. Why? Because it's the heat dissipation effect that it's pushing the thermogenesis, and that's what we see here when we place the animals in a chamber that I call the tropical la-la land, which is basically an incubator that keeps animals at 32 degrees, which for rodents it's a thermal neutral temperature. It's a temperature at which they don't need to engage activity of the brown adipose tissue. Then the weight loss effect is lost. So to make the long story short, here we have a graphical abstract that was first drawn by Sirenia Sketches during the first lockdown. And she was very inspired by the whole situation of staying at home that by now we're all fed up. She made this cartoon where we have the pegylated amphetamine with his little mustache, glasses, and a hat. And this is to pay reference tribute to a Netflix series called Breaking Bread, where you have Mr. Eisenberg, who was also cooking amphetamines in his kitchen. And Mr. Eisenberg here, what it's doing is that it stays away from the brain. And by staying away from the brain, we have the cardioprotective effect. So the heart is also whole happy because it stays away from the bad guy, Mr. Eisenberg. And Mr. Eisenberg here, pegylated amphetamine, drives weight loss because it's dialing up the temperature, so the thermogenesis capacity, while keeping the window open. So while keeping heat dissipation, active heat dissipation engaged. And this is actually an effect that it's speculated to exist also in drugs like clenbuterol, which is a beta-2 adrenergic agonist that unfortunately has been abused by illicitly for weight loss and by bodybuilders. And that's in the news, and there's papers on PubMed that express their concern on abuse of clenbuterol, which is a beta-2 adrenergic agonist. Now, the theory behind this is that we have a neuroendocrine loop of leptin action that has an afferent arm and an efferent arm, which are neurons. And the way to overcome leptin resistance, some have proposed that mitigating the efferent arm has an effect on the homeostasis. So the group of Philip Scherer has done that. He has demonstrated using mouse models in which he had using neutralizing antibodies against leptin or models of temporally controlled knockout of leptin. It demonstrated that by suppressing the leptin axis, by making the afferent axis smaller, there's actually a return towards homeostasis. And my proposal, it's the reciprocal of that, in which we facilitate the efferent axis so that we can restore homeostasis. So sympathofacilitators, therefore, they bypass central leptin resistance by amplifying the efferent sympathetic axis of the neuroendocrine loop of leptin action. And it needs to be noted that leptin, the circuitry that goes from the brain down the spinal cord, across sympathetic ganglia onto the adipose tissue, it's a circuit that does not encompass input onto the heart. And maybe also that's why we don't have any cardiogenic effects, because simply the heart branch does not belong to the circuit that underlies this neuroendocrine loop. All of this work was done by people that I have to be thankful to. Particularly, I would like to thank also my collaborators, Gonçalo Bernardes, who had the first idea of pegylating the amphetamine, Chris Glass at CSD, who was hosting Roxana for a summer, so that she did all the sequencing experiments there together with Jason Seidman. I have to thank also Elsa, who was a buddy of Roxana in the Nature Medicine paper. Inês Mayhu, who just started a postdoc at the Max Planck Institute for Metabolic Research in Cologne with Jens Browning. And there's a variety of people here. Some of them, they moved to Oxford. Of course, I have to thank the funding agencies and also my collaborators in Cambridge who helped us with the mass spec of the pegylated amphetamine. And some of these people, they moved to Oxford 2.5 years ago, where I started my lab. As you know, Oxford is the the birthplace of Harry Potter. And if you are interested in this sort of topics, please do not hesitate to contact me, and I would be happy to talk. Thank you so much.
Thank you, Anna. Thanks for sharing this beautiful work. I really appreciate your fresh perspective on the neuroimmune metabolism. And I also appreciate you sharing with us how we should keep our eyes open, right? And sometimes results are different from what we expected, and it's so important to think about it and readjust our hypothesis and keep on moving forward. So
I think that's the nature of research. And
I completely agree. And it's very important to take the time to reflect on that. So I really appreciate that too.
Yeah.
And that was a beautiful graphical abstract. I think it's the first one I see that is so dynamic.
So allow me to advertise the services of Sirenia. She's really a good. Oh, and I cannot push my slides backwards. Okay.
And good artist.
Sirenia, you can Twitter her. She's or just send me an email and I'll send you her contact. She's really good.
Thank you.
And she would be happy to take more jobs.
Okay, I know you're very interested in art as well. I should have commented on that in my intro. So thank you again. I think Elena is going to remind us, how can people ask you questions?
Yes, yes, definitely I will. And I want to join Carla thanking Anna for a wonderful talk and for breaking new ground in the field of neuroimmunology. Thank you so much, Anna. Okay, so if you'd like to ask questions to Anna, then go to the Twitter account Global IMMUNOTALKS, and there is already a tweet that says ask questions for Dr. Anna Domingos here, and Anna will reply to that tweet. For asking questions, you reply to that tweet with the hashtag Global Immuno, and Anna will be using the ImmunoSpeaker Twitter account for answering questions contingent with her availability. So thank you so much to everyone for joining with us again live, and for those that watch the talk in YouTube. And just a reminder that next Wednesday we'll have Pam Schwartzberg as our global immune speakers. So we hope to see you then. Okay, thank you so much. And
thank you, Anna, again.
Well, I'm sorry about the video. That's really unfortunate. Yesterday was working. So anyone who may want to ask me questions, they can call me up and we can do a face-to-face.
That sounds great.
Okay, great. Perfect.
Thank you. We could listen you perfectly and we could see all your slides.
All right.
Thank you, Anna. We'll be in touch. Take care.
Bye-bye. Adios.
Ciao. Bye.